Hello, everybody, and welcome to session number 2 of From the Ground Up Western Cover Crop Council's Pacific Northwest Subregional Conference Series. My name is Stephen Hines with the University of Idaho Extension, and I'll be your moderator today. Today, our session topics are going to discuss current cover crop projects in vegetable production. So I'd like to take this opportunity to introduce our next speaker. Nick Andrews is with Oregon State University Extension, and he's going to visit with us about cover crop nitrogen contribution and on-farm trials. Welcome, Nick.
The topic of my talk is cover crop nitrogen release and on-farm trials. I've been working with Dan Sullivan, OSU soil scientist, as a mentor for, I guess, 15 years now. And this is some of the work that we've been doing with funding from Western SARE and more recently the Tualatin Soil and Water Conservation District. So one of the big questions that we've been trying to work on is how much nitrogen, plant available nitrogen, is this beautiful cover crop going to provide after you incorporate it? So what's the fertilizer value? Unlike fertilizer bags that where you know your application rate, you have a guaranteed analysis. You have none of those things with a cover crop stand. So it's sometimes a little tricky to get to an accurate plant available nitrogen prediction. I'm going to go through these slides pretty quickly. Don't worry about catching everything. All of the information that I'm going to cover is in these two publications. If you just write down OSU and then those publication numbers, EM9235 and PNW636, you'll be able to download those pretty easily. They're free online. And then I'll also talk a tiny bit about this decision tool that Dan and I have developed. It's the OSU Organic Fertilizer and Cover Crop Calculators. So first of all, if you do want to sample your cover crops to estimate plant available nitrogen, you have to go out in your field and the most accurate way is to go out in your field and take some biomass samples. We use a 2 foot by 2 foot sampling frame, but you can use whatever size you want. We generally take 4 samples per field. It doesn't matter that much what size your frame is. Larger is a bit better than really small, but consistency makes it less likely that your numbers are going to get mixed up. So consistency is sometimes better than perfection. This was a red clover cover crop interseeded into butternut squash by Jim Bronick at Praying Mantis Farm. And that's in April. Beautiful stand in what would otherwise have been a bare field. He's going to talk a little bit about that. So when I'm sampling a field, I walk to the far end of the field and then walk back and try to find 4 kind of representative areas to take a sample. A couple of ways I take a sample. One is to work my frame through the stand and then cut the above ground biomass. Another is to knock down the cover crop and lay the frame on the thatch and cut through that thatch. That's really our preferred method for tall, tangled cover crops where we've got a lot of common vetch and oats or anything that's difficult to work your way through. Some people have built sort of U-shaped sampling frames out of rebar, which is a great idea. You can kind of shimmy that at the base of a tall and tangled cover crop. So that might be a good alternative to this laying it down and taking a sample. This was when we were doing the research. We would haul all of the samples back to our field lab and then mix them up. Now we just do this on the tarp in the field for advisory work, but you combine all 4 of those samples, all 4 of those frames, mix them up, get rid of any soil, weigh the fresh weight, and ask the lab, send us about a 1 pound subsample off to the lab and ask them to dry and grind the whole sample and analyze it. A lot of labs will want to take a tiny subsample out of your bag and dry and grind that. But if you've got mixed cover crop, especially if you've got mixed cover crops, mixed species, It's important, that's an important request, that they dry and grind the whole sample and mix it up before they take that little tiny subsample that they're actually going to analyze. All you need from them is percent dry matter and total percent nitrogen. It's about 20 bucks a sample. I haven't checked the price recently, but pretty reasonable. I think at this point you're sort of into it for about 45 minutes to collect your sample and bag it up. And then, you know, depending on how far away your post office is, and then a $20 fee plus shipping for the analysis. Make sure that you record the area that you sampled. So 4 square foot quadrats times equals 16 square feet per field, and then record the total fresh weight of your bulk sample, that whole bulk sample that Kristen is handling there in the photo on the left. If you don't have those numbers, you won't be able to make any calculations. So I'm going to just move quickly to the cover crop calculator. It's got 2 versions online, a larger farms calculator on using acre units, a small farms and gardens calculator using 1,000 square foot units. It predicts nitrogen mineralization from cover crops and organic fertilizers. It also has some cost estimates I'm not going to really get into. The cover crop cost estimates include management costs, not land costs, but it does include depreciation, fuel, labor, that sort of thing. And then the fertilizer calculator, part of it calculates macro and micronutrients supplied by fertilizer rates that you select. In 2020, we revised the calculator with new nitrogen predictions. So now the models predict nitrogen mineralization and nitrogen immobilization from fertilizers and cover crops. You can actually enter data for up to 8 cover crop fields at a time now. Jeff, I think we're assuming this, we developed this for Western Oregon, so you would definitely have to adapt it for conditions in Alaska. But the PAN predictions, this is the hard science work that Dan led. These are the formulas that we put into the calculator for predicting plant available nitrogen release after 4 weeks after incorporation and 10 weeks after incorporation. And that's how it matched up with our data. The blue chart is the 4-week mineralization predictions, and the red chart is the 10-week mineralization predictions. And you can see it where that arrow is, it's a segmented linear equation. And you can also see that with low nitrogen, it drops down below 0, which is that's really the very useful new feature. If you're interested in the scientific details, Dan Sullivan gave a very nice presentation at the Tri-Societies meeting last November, so you can find it there on their website. In our extension publication, we've got some lookup tables that if you are estimating or if you know the total percent nitrogen in the dry matter in your cover crop, you can look up mineralization rates, either using percent of total nitrogen units here or pounds of plant available nitrogen per dry ton of the cover crop. And this is the same information in a chart. And this is total nitrogen as percent in the dry matter. And then on the y-axis is the plant available nitrogen supplied in pounds per ton. So you would multiply these numbers by the ton of dry matter in your cover crops. So these tools this information is for people that want to use table values rather than sending their sample into a lab. So we've got a couple of SERP grant-funded projects. Doug is leading a research to grassroots grant, and then I'm working with Clint Taylor on this Tualatin and Soil and Water Conservation District grant. We've got on-farm cover crop trials that are going to contribute to an online database that we're developing with that Western Stair grant. And the idea is that we'll be able to share data with farmers and with the Cover Crop Council. The farmers in this study selected two objectives. They wanted to look at which cover crop will give them the maximum dry matter without immobilizing nitrogen. That's the bottom one there. And they wanted to look at which cover crop will give them the maximum nitrogen contribution for the following crop. We're also doing some nitrate monitoring and working with them on their fertilizer programs. In 2020, we have 5 farms. In 2021, looks like we've got 7 farms, and we're hoping to have 9 farms collaborating in 2022. So here's the results of the maximum pan study. These are all legumes, of course, 5 varieties, common veg VNS, 2 varieties of crimson clover. This is Jim Bronick's crimson clover. All the farmers in this study use that. And AU Merit hairy vetch and woolly pod hairy vetch. So this is total dry weight, including the weeds. You can see that there was quite a bit of variation on from the farms, but it's between 3,000 and 6,000 pounds per acre of dry matter from those legumes. This is the cover crop percent nitrogen, including the weeds again. You can see it looks like, so this is just 3 farms. Each farm is a replicate. These trials were unreplicated on each farm. So we're not doing any statistics yet because we're still collecting more data this year. So it looks like, initially maybe the AU Sunrise was a little bit lower on the percent nitrogen and the vetches were a little bit higher, 3.5% to 4% versus 2.5% to 3%, give or take. But this is preliminary. And then this is the cover crop total nitrogen, and I'll jump ahead to the bottom line, which is the plant available nitrogen. And these are the predictions that we got from the OSU calculator. So, but all of those factors in the previous slides contribute to this final fertilizer value estimate. And you can see that Crimson did really well on one farm. We had a lot of variability in establishment on the farms. The VNS common vetch was a pretty strong performer. And then the AU Merit and the woolly pod hairy vetch were also fairly strong performers. So along the y-axis, this is 60 pounds of fertilizer value. This is 100 pounds per acre of fertilizer value. Lots of nitrogen coming from these cover crops. So going to the maximum dry matter trial really quickly, the hypothesis was that cereal cover crops provide more dry matter, but farmers didn't want to immobilize nitrogen after they incorporated the cover crop. They didn't want the cover crop to rob, nitrogen from their vegetable crop. And that hypothesis looks like it was correct. We got about 6,000 to 8,000 pounds of dry matter from the cereal and vetch cover crops, compared to 3,000 to 6,000 pounds of dry matter for the vetch cover crops. The treatments here, because we wanted, as I mentioned, our key interest area was how much vetch seed do we need to put into this triticale stand in order to avoid immobilization. So you can see we've got 100 pounds of triticale everywhere, 0 vetch, 12 pounds per acre of vetch, 24 pounds per acre of vetch, and 36 pounds per acre of vetch. You can see the impact on the total nitrogen in the cover crop, quite a bit more in those treatments. The cover crop percent nitrogen was also higher, and we think the tipping point between immobilization and mineralization is somewhere between 1.5% and 2%. So we're kind of seeing what we would expect in these different treatments. And then this is the penultimate slide. I've got one more if I have time. The blue lines are nitrogen being immobilized 28 days after incorporation. The orange lines are bars are nitrogen being immobilized 70 days after incorporation. So the 0 vetch and the 12 pounds per acre of vetch treatments we're immobilizing a bit of nitrogen, especially in the short term. The 24 pounds of vetch looks like it immobilized a bit of nitrogen, 10 to 15 pounds per acre at 28 days, but by 70 days, it was actually mineralizing a little bit. So, and then the 36 pounds was actually mineralizing nitrogen from the beginning, and we actually got about 40 pounds of pan from that treatment. So we think that our sweet spot with 100 pounds of triticale is somewhere around 20 to 25 pounds of vetch to 100 pounds of triticale, but we'll get more results. We're also starting to do some work with interceding. This is just a plug for Doug Collins' seminar tomorrow. It's a Maritime Pacific Northwest no-till session, and I'll talk a little bit more about these. These are all just different types of interceding tools that we're playing around with. So I'll talk about that a little bit more tomorrow.
All right, thanks, Nick.